Good morning, students. In today's lecture, we will study the factors that influence the thermal destruction of microorganisms. Understanding these factors is very important in dairy processing because heat treatment is used to make milk safe for consumption while maintaining its quality. Let us first understand the basic concept of thermal destruction. Thermal destruction refers to the inactivation of harmful microorganisms by applying controlled heat. In dairy processing, raw milk undergoes heat treatment to achieve three main goals. First, to ensure microbial safety. Second, to extend shelf life. Third, to preserve nutritional quality. Thus, heat treatment converts raw milk into a safe and stable product. One of the most important factors is the type of microorganism present. Different microorganisms have different levels of heat resistance. Bacterial spores are highly resistant to heat. They have thick protective layers that allow them to survive even boiling temperatures. Examples include Bacillus and Clostridium species. In contrast, vegetative cells are much heat sensitive. They lack protective barriers and are destroyed easily during pasteurization. Examples include Salmonella, E. coli, and Listeria. Another important factor is the initial microbial load. This refers to the number of microorganisms present before heat treatment begins. When the microbial load is high, more microorganisms must be destroyed. Therefore, a longer heating time or higher temperature may be required. On the other hand, when the microbial load is low, standard pasteurization conditions are usually sufficient. Thus, starting microbial count directly affects the intensity of heat treatment needed. The composition of milk also affects microbial heat resistance. Milk contains proteins and lipids that can protect microorganisms from heat. Proteins may form protective barriers around microbial cells. Lipids or fat globules act as thermal insulators and reduce heat penetration. As a result, high-fat milk products may require more intense heat treatment to achieve the same microbial destruction. The pH of the medium also influences thermal resistance. Microorganisms show maximum heat resistance around neutral pH, which is approximately pH 7. When the pH moves away from neutral conditions, microorganisms become more vulnerable. For example, acidic foods require less heat treatment because microorganisms are already weakened by the low pH. Thus, pH plays an important role in determining heat treatment intensity. Fat content and milk solids also affect thermal destruction. Fat globules act as thermal insulators. They shield microorganisms from direct exposure to heat. Similarly, milk solids increase viscosity, which slows down heat penetration. As a result, high-fat milk products may require more intense heat treatment to achieve the same microbial destruction. Water activity is another critical factor. Water activity refers to the amount of free water available in a product. Microorganisms tend to be more heat resistant in low moisture environments. Dry conditions protect cellular proteins from heat. In contrast, high water activity makes microorganisms more susceptible to heat. Therefore, products like powdered milk may show different heat resistance compared to fresh milk. The heating method and temperature also influence microbial destruction. For example, HTST pasteurization uses temperatures around 72 degrees Celsius for about 15 seconds. This process destroys vegetative cells while preserving product quality. UHT processing uses much higher temperatures, around 136 to 145 degrees Celsius, for only a few seconds. This process achieves commercial sterility and allows milk to be stored at room temperature. The key principle is that higher temperature allows shorter heating time for the same microbial destruction. Often, multiple factors act together. When several protective conditions exist, microorganisms may become extremely resistant. For example, high fat content, neutral pH, and high microbial load can combine to create very high thermal resistance. This is called the synergistic effect. In such cases, standard pasteurization may not be sufficient and stronger heat treatments may be required. These factors have great practical importance in dairy heat processing. Proper heat treatment helps eliminate harmful pathogens and prevent foodborne illness. It also extends the shelf life of dairy products by reducing spoilage microorganisms. At the same time, careful control of heat treatment helps preserve nutritional quality and sensory characteristics. Thus, thermal processing protects public health while maintaining product quality. This slide summarizes the major factors affecting thermal destruction of microorganisms. They include type of microorganism, initial microbial load, composition of milk, pH level, fat and milk solids, water activity, heating method, and combined effects. All these factors determine how resistant microorganisms are to heat treatment. To conclude, thermal destruction of microorganisms is influenced by several interacting factors. Understanding these factors allows dairy technologists to design effective heat treatments that ensure food safety while preserving product quality.